First we will see introduction of the apoptosis. Apoptosis, it is a biochemical event in the body. In this, it is called as a programmed cell death. It is called as a programmed cell death because cell it regulates its own death, and the cell regulates its own death with the help of production of certain enzymes, and that enzymes are a proteolytic enzymes, and that enzymes bring about the degradation of the nucleic acid and degradation of the proteins in the body, and there is the formation of the apoptic bodies, and finally it will lead to the death of the cell. In body, there is always a balance between the formation of the new cells and previously existing cells. Imagine that if continuously new cells are formed in the body and the previously existing cells, they are remain as it is in the body. So what will happen? This will lead to a abnormal condition in the body, and for that the apoptosis is needed in the body. So we will see the history of the apoptosis. In that, scientist Carl Bogaert, he was the first to describe the process of apoptosis. Later on, Walter Fleming, he gave a more description about the programmed cell death. Later on, a group of the scientists, that is Sidney Brenner, Hurwitz and Sulstan, he awarded with a Nobel Prize for discovery of the genes involved in the apoptosis. And that genes involved in the apoptosis, they were discovered during the study of nematode, that is C. elegans, and the same genes were found to be present in the human beings that controls the apoptosis. Then the next point is how the apoptosis is triggered in the body. There are several mechanisms through which apoptosis is triggered. Some of them are infections, especially viral infections, then generation of the protein misfolding, and that protein misfolding due to mutation in the body, then DNA damage, and also radiation. Whenever there is exposure to the radiation or generation of the free radicals in the body or hypoxia condition in the body, that will lead to the generation of the apoptosis or stimulus of the apoptosis. Then the next point is need of the apoptosis. So why there is a need of apoptosis? Apoptosis is needed for a normal development of the body. Especially in embryonic conditions, it is required for formation of the fingers and toes. Then it is also required for formation of the proper junctions or proper connections in the neuron, between the neurons in the brain. Apoptosis, it is also needed to destroy certain cells. The cells that are not needed by the body, they get killed by the process of apoptosis. The cells, whenever they are not needed by the body, then a certain signal is carried to the cell that the cell is need to undergo apoptosis, or there is a need of killing of that cell. Then it is also required for apoptosis also plays a role in the adaptive immune system, that is T and B cells which are self-reactive. Whenever B and T lymphocyte, they have a self-reactive antigen, then that T and B lymphocytes, they are no longer needed for that body. So T and B lymphocytes which are self-reactive antigen, they get killed by the process of apoptosis. Then apoptosis, it is occurring at a high rate in the bone marrow cells where the most of the blood cells are formed. Apoptosis, it is essential in the embryonic development, that is development of the homeostasis. It is also required for maintenance of the steady population of the cells. So what happens if apoptosis is not there? Think about that. Here, if apoptosis is not there, then it will lead to the improper differentiation of the fingers and toes. So cell undergoes death by two mechanisms. In that, the first is necrosis and another is apoptosis. So necrosis means it is a cell death by injury. It is also called as a accidental cell death. And apoptosis, it is a cell death by suicide, that is a programmed in the body. It is not by accident. So there are several characters of the apoptosis and necrosis. This is a normal cell. In this normal cell, whenever apoptosis is there, there is a cell shrinkage occurs and a chromatin condensation occurs. After that, some of the morphological changes are there, like there is the formation of the surface blebs, and later on the surface blebs occur and a phagocytosis is there. So cell shrinkage is the important character of the apoptosis, while in necrosis the inflammation is the important character of the necrosis. There is the cell swelling occurs and finally bursting of the cell is there, and because of that there is the release of the intracellular components are there, and because of this there is the cell death occurs. So there are some points which are differentiating between the apoptosis and necrosis. Apoptosis, it is a non-physiological event, while apoptosis is a physiological event, while necrosis is a non-physiological event. Here there is the shrinkage of the cell is there, and in necrosis there is the inflammation of the cell is there. Then the surface blebbing occurs during the apoptosis, while here there is no any surface blebs form, and also there is no any loss of integrity of the plasma membrane in case of apoptosis, while in case of necrosis there is the loss of plasma membrane integrity is there. Then one of the important point about this is apoptosis, it is a ATP dependent process, that is energy dependent process, while necrosis is a passive process, it doesn't require any energy. Then the important enzyme which are involved in the apoptosis is caspases. The term caspases, it is the C stands for cysteine, which is present in the active site of the enzyme, and this cysteine which is present in active site, it brings about the proteolytic attack on the target protein. It brings about a attack on the nucleophilic attack on the target protein and bring about a proteolytic cleavage, and that cleavage occurs at a aspartic acid residue, therefore it is named as a caspases. Initially it is synthesized as a precursor, it is called as a procaspases, and later on these procaspases, they are converted into active caspase. Procaspase is a inactive form of this enzyme. Then the next point is types of the caspases. There are two types of the caspases. First is initiator caspase. In that initiator caspase, 4 different caspases are involved: caspase 2, caspase 9, caspase 8 and caspase 10. So it is called as an initiator caspase because it initiates the cascade. Then the second type is effector caspase, which is also called as an executioner caspase. In this, 4 caspases are involved: caspase 3, caspase 6, caspase 7 and caspase 14. So what are the morphological changes present in the apoptosis undergoing cell? First and important is the cell shrinkage. There is the reduction in the size of the cell, that is cell shrinkage occurs. Then nuclear chromatin, it undergoes shrinkage and also condensation. They condense at the center or the periphery. That process is called as a pycnosis. Then chromatin undergoes karyopexis, that is it disintegrates. It disintegrates and fragments. Then the cell surface, it starts forming a blebs on the surface, and then they break into a fragments, and that fragments are called as a apoptic bodies. Then the cell surface is chemically altered. That chemical alteration is there so that it can be readily engulfed by the phagocytes. So in this diagram some of the morphological changes are shown. First, this is a normal cell. In this normal cell, the cell shrinkage is there. Then there is the formation of the membrane blebs. Later on, the nuclear collapse occur, and finally there is the formation of the apoptic bodies, and that apoptic bodies then undergo lysis. So this is the process of the apoptosis. Apoptosis is a highly regulated process. So this highly regulated process is controlled by some of the factors in the body, and that factors are anti-apoptic factors and pro-apoptic factors. Anti-apoptic factors, that is the factors which are against apoptosis. They control apoptosis, that is they inhibit the apoptosis. And pro-apoptosis factors means the factors which promotes apoptosis, which stimulates the apoptosis. So the examples are BCL2 family proteins. These BCL2 family proteins, they are the conserved proteins that share a homology domain. These BCL2 family proteins, they fall into two categories. First is anti-apoptic proteins. Examples are BCL2 and BCLXL. And pro-apoptic factors are BAX, BAD, PIC, BEAM, PUMA and NOXA. These are the factors that promote apoptosis. So you have to remember these factors because they are very important. Then the next point is pathways of the apoptosis. In that, the two pathways are there. First is intrinsic pathway and second is extrinsic pathway. So first pathway is intrinsic pathway, which is also called as a caspase dependent intrinsic pathway, or which is also called as a mitochondrial pathway or mitochondrial apoptosis. It is also named as a mitochondrial mediated cell death. As the mitochondria plays important role in this, it is called as a mitochondrial mediated cell death or mitochondrial apoptosis. Then the extrinsic pathway is the another pathway of the apoptosis. So in this video we will see only intrinsic pathway. So intrinsic pathway, for that a internal reason is there, that is internal DNA damage is there. So they require a receptor. जरा stop करते